Yo, today I guess we're just gonna go through I think just an overview of DNA rep. Um it's I don't think it's that deep I mean from what I understand it's not that deep to know on the MCAT, but I guess we'll only focus on eukaryotes because well I haven't learned about prokaryotes yet. I don't want to tell you the wrong information. I will say there's a distinction between eukaryotes and prokaryotes because since the eukaryotic genome is far larger than the prokaryotic genome, one difference is that eukaryotes have more than one origin of replication, whereas prokaryotes usually just have one. An origin of replication. Dang, I can't spell. Origin of replication. This is nothing but where replication begins. Now, like I'm saying, right, eukaryotes can have many origins of replication because there's so much more to replicate, right? So for example, this is gonna be a really stupid drawing, but like say we have, you know, DNA, right? This is prokaryotic DNA. Holy crap, this is a bad drawing. An origin of replication, for example, here, you know, when it is time to replicate, this is what would happen. It would enlarge in that area and say this is 5 to 3, 3 to 5, and you would just start replicating. Now, for eukaryotes, um, if we had, same DNA or whatever, blah, 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 there would be multiple humps. And so that means that replication can start here, and just imagine it's the same, like, whatever. Replication can start here, replication can start here, replication can start here, here, right? So that means that, or it basically is like a way for eukaryotes to be more efficient with how they replicate. That's pretty much it. Okay, now let's go through the exact mechanism, right? So DNA is whatever it's called, anti-parallel, which just means there's one strand that goes from 5 prime to 3 prime, and the opposite strand goes from 3 prime to 5 prime. And always remember that DNA replicates in the 5 prime to 3 prime direction. So yeah. All right. So there are a couple of there are a couple of enzymes that are important to know. Um, so helicase, how can you even see that? Helicase is one of them. If I can freaking helicase. So say we have, we have our base pairs, whatever. Okay, imagine I'm doing a better job here, right? And the way how DNA kind of like the two strands of DNA stick together is through hydrogen bonds, right? So basically, what helicase does is it cleaves or it breaks those hydrogen bonds. Okay, so that's able to separate the two strands from, like their connections. Now, obviously, that still does mean, if helicase is not there after it's been broken, the complementary strands could just like link up again. So for to prevent that from happening, we have other proteins that are on the backbone. These are single stranded binding proteins or something like that. Just know it's something. Basically, this prevents it from going back or from hydrogen bonding again. So therefore, the DNA replication could actually, happen. If it's bonded together, then you can't do it. All right, let me redraw it. So now we understand what helicase is. I think it's also important to understand that, DNA is coiled. In this picture, the stupid picture I've drawn, it's a little bit hard to understand, but it looks more like this. And because of that, there's a lot of torsion, a lot of tension in this structure. So, and that kind of like it causes like like electrophilial or whatever it's called, electrostatic interferences and other like steric interferences, whatnot. So there's another important enzyme, topoisomerase. And what topoisomerase does is it basically goes upstream and essentially reduces the torsion. Now, exactly how it does it is it actually like snips the DNA and then releases tension and then rejoins it. So yeah, that's basically it. What I think of, it's like, I don't know what's the best way to say this, but like I think of topoisomerase as you're blowing into a bubble and then you're slowly releasing the pressure in the bubble, or in a bubble. No, not a bubble, a balloon. You're like slowly releasing the pressure on the balloon before it like goes back to its resting state. And I just wanted to clarify one more thing that just popped into my head. This thing of the strands coming back together, um, it's a process, or I mean, there's a name for it. It's called annealing. Don't forget, whatever. Not that deep, I don't think so. So, anyways, we got our 5' strand. We have our our origin of replication, 3' strand. We have our 3' strand here, and okay. Now, like I said, um, DNA is replicated from the 5' to 3' direction. So what happens is, like, if you were to like, break this down into smaller chunks, this would technically be the relative 3' strand. This would be the relative 5' strand. And what's happening is replication goes this way, 5' to 3'. And similarly, since this is anti-parallel, it goes 5' to 3'. Now, obviously, that makes sense, right? You will just continue in a smooth fashion down this way. You'll continue in a smooth fashion down this way. However, this area here, you're going from the 3' to 5', right? So instead of it being like a smooth like transition, it's going to be in small like fragments of DNA that is replicated, right? And these are the relative 5 to 3. Okay, these are called Okazaki fragments. I guess the important thing to know is that on the side of the 5' of the DNA is synthesizes one strand, which is called the leading strand. And on the 3' half of DNA, the lagging strand, DNA is replicated in Okazaki fragments. All right, that's looking it. Couple more things to start the actual process of replication. We require a primer, like an RNA primer, that allows polymerase to bind, and that primer is called RNA primase. And as the name suggests, this is an RNA, like code or just a strand of RNA that allows for what we're going to talk about now, DNA polymerase, to proceed through for replication. Speaking of DNA polymerase, there's a couple of different DNA polymerases. The one that's important in actually synthesizing or replicating is called DNA polymerase 3. So this synthesizes the complementary strand. And well, yeah. So DNA polymerase will bind here and work its way down. Choo choo. I'll go. Cool. But if you notice, we're gonna have a new DNA strand. Well, we're still gonna have an RNA primase over here, and this is gonna be that replicated strand. So we want to then replace this with DNA because obviously, you can't have RNA and DNA. And this is what DNA polymerase 1 does. And what does it do? Like in writing, replaces the primase RNA strand. The way I like to remind or to like remember this is primase. Holy crap! I cannot. I just cannot type. I'm not even typing, bro. I cannot write. Primase starts with this prime. I think primary. You know, first, and it's DNA polymerase 1. So yeah, I guess that's like the basics of DNA